If you or a loved one is living with cancer, you probably have two focal points: how to rid yourself of it and how to prevent it from coming back. You'll get some terrific information today on Cancer Concepts and Complements with Dr. James Belanger. We'll explore natural medicines which may enhance conventional therapies, share cutting-edge research, and offer insight on how diet and natural medicines can play an important role in treating and preventing cancer. The information presented herein is in no way intended as a substitute for medical counseling. Now, here is Dr. James Belanger.
Hello, everyone. Welcome back to Cancer Concepts and Complements. I'm your host, Dr. James Belanger from the Lexington Natural Health Center in Massachusetts. If you didn't catch the last two shows, we've been talking about the immune system and cancer. And last week's show, I talked about various types of suppressor cells, cells that actually can surround the cancer and protect it, that are derived from our own body, from our own immune system. So just to recap, cancer cells are not just sitting there all by themselves. They are surrounded by a network of cells and proteins. Those cells can include cells that are trying to kill it, like killer T cells and natural killer cells, and cells that are inspecting, dendritic cells. But they can also be surrounded by cells that actually are protecting it. And of course, the immune system wins the battle if there's more of those killing cells around the cancer than the ones that are protecting it. And those protecting cells can be called suppressor cells, and they include various types of cells. One's called the myeloid-derived suppressor cell, or MDSC. Then there is what's called the regulatory T cell, which I'm going to talk a lot about today. Then there's the neutrophils and what's called the Th2 cells. And then there's cells that are called cancer-associated fibroblasts, which make what's called the stroma. And I mentioned the stroma when I was talking about chemotherapy in some previous shows. And that is what protects the cancer cells against chemotherapy, causing blood vessels to collapse around the tumors. And it also protects them from the killer cells, the stroma, the cancer-associated fibroblasts. And so today we're going to expand more on some of the other suppressor cells. And one of those other suppressor cells is called the neutrophil. I talked a little bit last time about it, but let's talk about it some more. Neutrophils are the most dominant white blood cell in our immune system. And the reason for that is they protect us against bacterial infections. We have a lot of bacteria in our body, on our skin, and in our gastrointestinal tract, so we have to be protected by it. So we have a lot of these neutrophils. But neutrophils, in terms of cancer, might not be good in excess. They actually make growth factors that help contribute to blood vessel growth and the production of the tumor stroma. Neutrophils make something called oncostatin M. They make something called VEGF, which I've talked about, Interleukin 8, and MMP9. And they contribute to the formation of these cells called the regulatory T cells, which suppress the killer cells. And so there's these different compounds that are produced by the neutrophil. One is called oncostatin M, and oncostatin M is used for various things, but one of its things is it helps in the forming of the stroma. There's this stuff called fibrinogen that's found in our blood, and it is commonly elevated in people that have cancer. And fibrinogen turns into something called fibrin around cancer. And fibrin is the same stuff that's found in a blood clot. So basically this fibrin gets produced around the cancer and causes blood vessels to collapse, which leads to low levels of oxygen, which interferes with basically the chemo delivery. It induces blood vessel growth, does a lot of different things. And oncostatin M is an inducer of this fibrinogen stuff. And it comes from neutrophils, excess neutrophils, and it can be measured. VEGF I talked about is really important for a lot of things. It helps form another suppressor cell called the myeloid-derived suppressor cell. And I mentioned last week measuring the VEGF and doing things to lower it to normal levels so those myeloid-derived stem cells or suppressor cells don't accumulate. It also is needed for the formation of the stroma. VEGF causes blood vessels that are feeding tumors to become leaky. And when blood vessels leak, proteins deposit around the cancer that protect it from the immune system. So that's also comes from neutrophils. And then there's other two things. One's called Interleukin 8 and one's called MMP9 that also come from neutrophils, which I'm going to get into in a second. But there is research showing that if neutrophils are too high in the blood, disease-free survival might not be as good after 5 years. For example, in the journal Cancer in 1976, they took women with breast cancer and they just measured their neutrophil count. And they noticed those that were over 5,800 in the neutrophil were less likely to be disease-free at 5 years. 38% were disease-free in 5 years if they had that higher neutrophil count. Those that had neutrophils of less than 3,000 had a disease-free survival at 5 years of 58%. So higher disease-free survival. Why? Because if there's too many neutrophils, they can start accumulating around microscopic cancer cells and help make these growth factors that lead to the formation of the stroma. And the stroma protects against the killer cells and the formation of a suppressive atmosphere. And so there's other studies. There's one where you can measure neutrophils and make sure that they're not too high. And I'm going to talk about natural things that might help normalize the neutrophils so they're not too high, but not too low either. But first, some of the compounds from neutrophils can be measured as well. Interleukin 8 is a interleukin, not to be confused with interleukin 6 and interleukin 10 and the other ones I've been talking about. But interleukin 8 is a compound released by neutrophils and other cells, and it's important for blood vessel growth in tumors. So if there's excess interleukin 8, more blood vessels can form in the tumors and can help them expand. It's also needed to increase the permeability of the blood vessels, making them leakier, which leads to the stroma developing around the cancer, which then protects it from the killer cells. In the journal Clinical Cancer Research in 2004, they measured serum interleukin 8 in women with metastatic breast cancer, and if it was elevated, the survival was only about 300 days in women that had it elevated. Those that had it at normal levels, survival was more than 750 days. So just that one thing can be measured, and you can do something about it. You can lower it, which I'll talk about in a sec. So you don't want to ever see elevated levels of interleukin 8 in the blood. It might reduce survival, and does it because it helps with those blood vessels, and it's from the neutrophils. So a good test to monitor and something that can be manipulated with natural medicines, which I'll talk about. MMP9 is another thing that comes from those neutrophils. It's called MMP9. It is an enzyme, which we're going to later talk is really important for that cancer cells use to remove things that are allowing it to be attacked by the killer cells. Killer cells come in to the area where the cancer is and release compounds. And in order to hide from the killer cells, cancer cells use these compounds called MMPs, matrix metalloproteinases. They're enzymes that actually break down surface receptors, confusing our killer cells so our killer cells can't see it, helping them send out decoys, should we say, so the immune system can't see it. Well, MMPs can be produced directly by the cancer cells, but they can also be produced by the neutrophil, and then that's another way the neutrophil leads to immune suppression. So you can measure some of the MMPs. There's different ones. There's MMP2, MMP9, MMP10, and MMP9 is available at a lab called LabCorp. And there's research in the International Journal Cancer in 2003 in Argentina, where they measured the MMP9 in the blood of 46 breast cancer patients over a 38-month follow-up. And they attained levels before surgery, a month after surgery, and then every 3 months over the course of that 38 months. And those that had normal levels of MMP9 over the 38 months never had any kind of reoccurrence. Those that had elevated MMP9 activity had some kind of reoccurrence within 8 months after the surgery. Why? Because MMPs are needed for expansion of the cancer, so it can expand out, and it's needed to remove these things that are needed for our killer cells to recognize the cancer. So we need to measure MMP9 and other MMPs in the blood of people and give things to help reduce those to healthy levels. And there are natural things that do that. But first, so a common drug used in people with cancer is Neupogen and Neulasta. It is a drug used to keep the neutrophils at a normal level. And it's a great drug. I'm not against that drug at all. We need neutrophils or we're going to get bacterial infections. And chemotherapy can lower our white blood cells and lower our neutrophils, put it as risk for bacterial infections. And Neulasta and Neupogen has allowed people so they don't get bacterial infections when they go through chemo. But if it's overused and overdosed, then the neutrophils can get much higher than that 3,000 mark that I mentioned, even higher than that 5,800 mark. And if that is something that's occurring every time there's chemo going on, where there's an overdose of the Neulasta or the Neupogen, then there could be starting to get an accumulation of those neutrophils, at least in different periods around the cancer, which might not be good. So just something to monitor, and they can adjust the dose so it's not too high for people, and even not even using it if the neutrophils don't go too low. So that's something that shouldn't be used, recklessly. There are some natural things that can raise neutrophils, not as aggressively as Neulasta and Neupogen, but they have been studied. And there's one, it's called berbamine, B-E-R-B-A-M-I-N-E. There's a study done, I don't have that written down, I think it was in the Journal of Ethnopharmacology, but there's a human study done where they gave this compound, which is found in plants like Oregon grape and golden seal. It's not berberine, it's called berbamine. Well, they gave this individual compound to people undergoing radiation and chemotherapy, and it helped keep their neutrophils at a healthy level. And I've used that many times. It definitely is not as aggressive as Neulasta and Neupogen, but at least can help spare people from needing those injections. The Neulasta and Neupogen injections are great, but some people get, bone pain from it, from inducing their bone marrow so much, and that can be painful. And then you get these large fluctuations in neutrophils from really high levels, and then they go down. Whereas this berbamine has a more gentler effect on neutrophils. It's been studied in people in China, so there's human studies on it. It's well tolerated, something to look at more. But another way to help with the neutrophils is to raise the lymphocytes. So there's two main categories of white blood cells in the blood, neutrophils and lymphocytes. And neutrophils are the dominant white cell in our blood. They represent over 50% of our white cells. But the other main group of white blood cells are the lymphocytes, which I've been talking about. They include the T cells, the B cells, and the killer or natural killer cells. And they're important too. And the other thing about Neulasta and Neupogen is they so aggressively raise the neutrophils that often the lymphocytes drop because the bone marrow is starting to produce more neutrophils instead of the lymphocytes. And we want a lot of lymphocytes. Lymphocytes are a lot of times the cancer-fighting cells that include the killer cells and those T helper 1 cells and the B cells that make antibodies. And I've mentioned in some previous shows that measuring lymphocytes, and there's been research done in breast cancer patients, for example, that if those lymphocytes are less than 1,000 cells per cubic millimeter, the person has a higher risk of getting a reoccurrence. So we don't want to raise neutrophils so high that the lymphocytes end up dropping too low. And so this copamine or this berbamine is something that helps raise the neutrophils, but not as aggressively, so that it keeps the lymphocytes at a decent level. But measuring lymphocytes is important. You want to make sure that at least there's a count of 1,000, or some labs it's expressed as 1 × 10⁻³. And there are natural compounds that can boost up the lymphocytes, and thereby then the neutrophils go down a bit, so they're not as high. They're kind of inversely related. Thymus extracts. I mentioned that before. Thymus is a gland in our chest, and it starts to wither away as we get older because we don't really need it as much as we get older because it's responsible for maturing and educating our lymphocytes. But when you're undergoing chemotherapy or radiation and stuff, we need the thymus gland, but it's not as active. So there's research showing that you can take thymus extracts from a calf. Calves have an active thymus gland, and there's compounds called thymosin and thymopoietin in these thymus extracts that help our bone marrow make more of those T cells and make more of those killer cells and things. So it's a useful thing if somebody's lymphocytes and T cells are starting to fall too much. There's another compound. It's an herb called convolvulus. It's also called bindweed. And this weed, kind of behaves like cancer. It spreads and invades and stuff, and that's why they call it bindweed, binding to a plant and like strangling it. And there's research done in a journal called PR Health Science Journal in 2002 in Kansas, where they gave mice this convolvulus extract or bindweed extract, and noticed it was a 70% inhibition of tumor growth in mice compared to a control, and the lymphocytes increased in production by 35 to 46%. So it helps boost up those cancer-fighting cells, and then the neutrophils often come down in an inverse relationship, but not too low, but enough so it's not over that 3,000 mark for the neutrophils. Then there's also you want to do things that lower the interleukin 8 if it's elevated, things that lower the MMP9, the VEGF, the things that are released by the neutrophils and other cells that contribute to those blood vessel growth and the stroma. So interleukin 8 is something that can be measured in the serum. It's available at a lab called LabCorp, which is all over the country. And if it's elevated, there are various natural things that have been studied to lower that. There are 2 B vitamins studied in breast cancer patients. One's called niacin, which is vitamin B3, and one is called riboflavin, which is vitamin B2, combined with something called coenzyme Q10. They used these 3 things together in a study in women with breast cancer and found that the interleukin 8, if it was elevated, was reduced. And I mentioned that's really important because there's a study showing that people live longer when the interleukin 8 stays at a normal level. Here's these 2 simple B vitamins plus some compound called coenzyme Q10, which is a natural compound made in our liver, but you can take it in a pill that helps lower this interleukin 8. So that's a simple thing to do. There's other things that lower interleukin 8, such as a mushroom called reishi, which I'm going to get into, which is great for helping with natural killer cells and helping with that. So various compounds that can lower interleukin 8. I mentioned previously that for VEGF, if VEGF is elevated, green tea is excellent for lowering that. This is where probably green tea, its main thing it affects. And so if somebody tests high for VEGF, vascular growth factor, in the blood, a green tea supplement that's concentrated for something called EGCG can help make that work better. And then MMP9. MMP9, we mentioned, is something that can be elevated, and it's responsible for the invasiveness of the cancer, helping it spread places, and it helps protect against the killer cells. And MMP9 can be measured in the blood, and if it's elevated, there's all kinds of things that lower it, but one of my favorite things is Grapeseed Extract. So these compounds found in grape seeds, they call them OPCs. OPCs is this compound found in the seeds of grapes. It's also found in other things like blueberries and all kinds of berries, have been studied to reduce MMP9. And so it's something that's easily given, well tolerated, and can help reduce the invasiveness. And like I mentioned, there's that study in Argentina where they just measured that, and if that was elevated, women with breast cancer are more likely to get a reoccurrence. Simple, well tolerated thing that needs some more studies. Well, okay, we're going to take a break now. When we get back, I'm going to talk about another suppressor cell called the regulatory T cell. This is Cancer Concepts and Complements. I'm your host, Dr. James Belanger.
This is Cancer Concepts and Complements with Dr. James Belanger. The information presented herein is in no way intended as a substitute for medical counseling. If you would like to find out more about the Lexington Natural Health Center, please visit lexingtonnaturalhealth.com. That's lexingtonnaturalhealth.com. Now, back to this week's program.
Hello, everyone. Welcome back. Cancer Concepts and Complements. I'm your host, Dr. James Belanger. Now we're going to talk about another suppressor cell. and it's a very important suppressor cell. Lots of research studying this. It's called the regulatory T cell. It is a type of T cell that is found in everybody's body, and it helps keep our killer T cells and our T helper 1 cells from stopping or causing autoimmune disease in our body. So if we didn't have these regulatory T cells, our immune system would attack our whole body. So they're important for that. But cancer cells like these regulatory T cells because they stop those killer cells from attacking them. So they actually mutations that develop in cancer cells can cause them to start producing these compounds called chemokines. Chemo for like chemical, kine is an attractant, a chemical attractant that helps attract regulatory T cells to their vicinity. So it brings on the cells that it wants to help protect it. And so it releases this something called chemokine number 17, CCL17, and CCL number 22, chemokine 22, and that binds to regulatory T cells in the vicinity and kind of drags them into the area. And one of these CCLs, the CCL17, is produced by those neutrophils that I just talked about before the break. Those neutrophils start collecting around the cancer. They can release a chemical, CCL17, which then drags in the regulatory T cells, the T-regs, they call them for short, and that helps surround them. So you can measure these chemokines in the blood. You can measure CCL17, and research in American Journal of Hematology 2012 showed that elevated levels of that before and after chemo was associated with a worse prognosis in patients with Hodgkin's disease. And so you want to measure that and make sure that's normal. I'm going to talk about natural things that can help reduce the T-regs. So they call them T-regs for short, to help keep those from accumulating. You can measure CCL22, the other attractant, and that's also been studied in Hodgkin's disease. Elevated levels are associated with more advanced stages of Hodgkin's disease, and that's been studied in British Journal of Hematology. So there's 3 main types of regulatory T cells. One is called the FOXP3 positive regulatory T cell. And then there's one called the TH3 cell and the TR1 cell. So there's basically 3 types. And one of the types, the TH3, produces an excess amount of something called TGF-beta, which I've mentioned before. TGF-beta, transforming growth factor beta, is one of the major contributors to the stroma development, besides the VEGF that I mentioned earlier. And that is the thing that surrounds the cancer, protecting the cancer. So we don't want that. TGF-beta is also really responsible for preventing the dendritic cell, those inspector cells, from communicating with the T helper 1 cell and activating the T helper 1 cell. So if TGF-beta is high, messages are never sent that the cancer is, somewhere in your body, and that's how it hides from the good killer cells and stuff. The TR1 cells secrete another compound called Interleukin 10, which I mentioned is responsible for helping to produce another suppressor cell called the myeloid-derived suppressor cell, and is also responsible for preventing the dendritic cells and the T helper 1 cells from becoming in good contact with each other and becoming activated. So this is why, people get cancer, and it's never detected by the killer system, and it then keeps growing into a big enough mass to be detected, and then it starts to spread places. And if this is not corrected after chemotherapy or radiation or surgery, and there's still some cancer cells present and they still have these suppressor cells around them, then they just do the same thing that they did before and they start to come back. So it's a key to measure these suppressor cells and start making some changes, and using the lab to help determine that you have made a change. So you can measure CCL17, CCL22, and you can even measure the regulatory T cells. But first, how do the regulatory T cells stop the immune system from attacking it? How do they stop the killer T cells and the natural killer cells from attacking? Well, one, they prevent the whole inspection process from taking place in the first place because they produce that TGF-beta and that interleukin-10. And those are key things that stop the dendritic cell, which are our inspectors, from coming in contact with our T helper 1 cell and activating it. And without the activation of the T helper 1 cell, there's no messages sent that there's even cancer in the body somewhere. So the killer cells might be sitting there. They're very good at killing, but they never get the message. So I talked about the inspection of your house, and then that inspector says you have a problem with the roof, but you never get on the phone and call somebody to, come by the house and fix it. Why? Because somebody stopped that signal. TGF-beta, interleukin-10 stopped the message from taking place. Another thing that the regulatory T cells can do to stop the activation of the killer cells, killer T cells and natural killer cells, is depleting a necessary amino acid from them. There's something called arginine. Arginine is an amino acid. It's a compound found in all protein. So anytime you eat protein, you get lots of things. One of those things is called arginine. And our T cells need this arginine. Without arginine, they can't proliferate, they can't, replicate themselves and start to mount a large army to attack. And there's research showing that when arginine is added to somebody, whether a healthy person or somebody with cancer, that the T cells become more active. The killer T cells, the helper 1 T cells, they can proliferate, they can expand. To help, mount a response to cancer. So this arginine is an enemy of the regulatory T cell, should we say. And so the regulatory T cell produces something called, or activates the production of something called arginase in cancer cells. Arginase is an enzyme that breaks down arginine. So any arginine that's in the vicinity of the cancer gets broken down by arginase, which is induced by the regulatory T cell. And so then any killer cells that are in that area don't have that arginine that they need to help them, kill and migrate into the cancer and expand. And so that leads to depletion of them. And then what something is removed from the T cell or not produced called CD3 zeta. CD3 zeta is needed on a T cell for it to start to proliferate and expand. And that is depleted when arginine is depleted. So there's research showing that arginine actually might help the immune system work better against cancer. There's a study done in the Annals of Surgery in 1990 in 420 mice. They gave them this cancer called neuroblastoma, and then they gave arginine. 1% of their diet was arginine, and they noticed that tumor growth was reduced, and they had better survival times compared to a group not given arginine. And how did it work? It enhanced those killer T cells. There's been studies done in people with colon cancer where they just gave arginine for 3 days before surgery. And then when they took the tumors out, they noticed there were more natural killer cells in the tumors and more killer T cells, and less infiltration of like regulatory and suppressor cells. So something that needs to be studied more. I mentioned arginine has also been shown to make chemotherapy work better. There's a study done in women with breast cancer. So it's something that has some dual effects. It might make chemotherapy work better and also make the immune system work better. They have just done these short-term studies where they just gave it for 3 days before. So it might be best just kind of given in a pulsed fashion where you just take it for 3 days, 3 to 5 days, then you stop it for a little while, then you go back on and off of it. And certainly you might not even need arginine as much if you reduce those regulatory T cells, which are causing the depletion of the arginine in the first place. So you can measure regulatory T cells in the blood. The Mayo Clinic has that test, and I use LabCorp has a contract with Mayo Clinic, and so I draw blood in the office and I send it to the Mayo Clinic, and they have a test that measures these 3 different types of regulatory T cells to get an idea if they're elevated or not in the blood. So that's one way of determining if regulatory T cells are elevated. Another thing is to measure the compounds they produce, TGF-beta, IL-10, and making sure those aren't elevated. And if they are, then lowering that. So there's direct ways and indirect ways of determining if a person is starting to form too many of those regulatory T cells. But there is a study done in a journal, Collegium Anthropologicum in Croatia in 2011, where they looked at people with colon cancer, and they noticed that they had an increased percentage of regulatory T cells in their blood. And the more advanced the stage of the colon cancer, the more likely they were to have elevated levels of regulatory T cells. A study done in the journal Pancreas in 2012 in Japan, 100 people with pancreatic cancer, same thing. The more advanced the stage, the higher the regulatory T cell counts in the blood. But then they also looked at survival. Notice that survival was lower in patients that had higher regulatory T cell counts than those that had normal levels of them. We all have some, but so if it's too high, that the 1 and 3 year survival in those that had the high regulatory T cells was 64% for 1 year survival, 30% for 3 year survival, versus 90% 1 year survival and 58% 3 year survival. So that's just survival for regulatory T cells. So what are some natural things that have been studied to reduce regulatory T cells? One of them is a mushroom, shiitake. Now, there's different parts of the shiitake. There's the fruiting body, the part that you see, the mushroom part. And then there is the root system of the shiitake called the mycelia. And the research that has been done on the mycelia, so not the fruiting body. Eating the mushroom might not help the regulatory T cells or an extract from the mushroom part, but the mycelium may. Studies done in Japan, Cancer Science Journal 2011, took mice with melanoma, and they fed them this shiitake extract, mycelium, and then noticed that the tumors were 1/3 the size after 21 days of the treatment compared to the group that didn't get the shiitake at all. And when they analyzed the amount of these FOXP3 regulatory T cells in the blood, they noticed that they increased in the animals with the melanoma that were not given the shiitake, but did not increase and actually decreased in the group given the shiitake mycelium. They also took the lymph nodes that were draining the area where the melanoma was and found a reduced level of regulatory T cells in the shiitake group, but not the group not given shiitake. And they also noticed that a compound released by the regulatory T cells called TGF also went down. So shiitake is a very useful thing, and I've seen it. You can measure the regulatory T cells with that Mayo Clinic lab. You can measure TGF, and shiitake mycelium extract can be very useful for lowering that. If there isn't, there's other things. There's another plant, a compound, natural compound, neem. Neem is a tree, grows in places like India, and the leaves and the roots and different parts are used, but the leaves are the things that have been studied to help possibly the regulatory T cells. Some really exciting stuff going on, studying compounds from the leaf. There's research done where they're using extracts from the leaf, or they also extract some compound from the neem leaf called neem glycoprotein, and found a lot of research on that. So there's one journal called PLOS Journal in 2013. They gave mice a sarcoma, and then they measured these compounds from the regulatory T cells, the interleukin 10, the TGF, and then interleukin 6 and other things. And they also measured the things that you want to have a lot of, like that interferon gamma, that thing that helps improve killer cell activity that's produced by the type 1 T cells. And they also even measured those chemokines, that CCL22, the thing that attracts the regulatory T cells. Well, after giving so many treatments of neem to these animals with sarcoma, they then looked at the types of T cells that are around the cancer and the expressions of these compounds from the regulatory T cells. and the interleukin 10 was reduced, the TGF was reduced, the CCL22, the thing that attracts it, was reduced, and the actual numbers of regulatory T cells were reduced. And then what's great about it was there was an increase of the killer T cells around the cancer, CD8 positive T cells. And we're going to talk about in a second that there's this thing called the FAS ligand. It's something that cancer cells express to cause cell death of killer cells so they can't attack them. Well, it reduced the expression of that in the cancer cells. So NEEM has many effects on the microenvironment, the environment surrounding the cancer, switching it from a tumor-promoting environment with lots of regulatory T cells to a cancer-killing environment where there are these CD8 killer cells and less of the suppressor cells. And then another thing that I'm going to talk about later is it increased the levels of what's called perforin and granzyme B in the T cells. These are things that the T cell uses to basically kind of poke holes and destroy the cancer. And sometimes the cancer cells produce things that reduce these compounds in the killer T cells. So the killer T cell, even if it's in there, has a harder time killing them. Well, NEEM extracts given to mice with sarcoma in this particular study, the T cells had higher levels of these compounds that destroy the cancer, perforin and granzyme B. And part of the mechanism is that the regulatory T cells reduce these compounds in the killer T cells. And then the third thing that helps reduce regulatory T cells that has been studied with some really great results is a compound from a plant called the white peony plant. White peony, not red peony, but white peony, is something called methyl gallate, where they gave them also to animals in the study Immunology 2010, and they measured these T regulatory cells in tumors to see if it affected them, and noticed that they were significantly decreased, and this led to reduced tumor growth and improved survival of the animals. So three major compounds that's showing a lot of promise to help affect these regulatory T cells: shiitake, mycelium, neem leaf, and methyl gallate. Okay, we're going to take another break, and when we get back, we're going to talk about some more compounds released by these regulatory T cells. This is Cancer Concepts and Compliments. I'm your host, Dr. James Belanger.
This is Cancer Concepts and Compliments with Dr. James Belanger. The information presented herein is in no way intended as a substitute for medical counseling. If you would like to find out more about the Lexington Natural Health Center, please visit LexingtonNaturalHealth.com. That's LexingtonNaturalHealth.com. Now, back to this week's program.
Hello, everyone. Welcome back to Cancer Concepts and Compliments. I'm your host, Dr. James Belanger. So before the break, we were talking about regulatory T cells, a type of cell that surrounds cancer cells, protecting it from the killer cells. And you can measure these in the blood. There's a Mayo Clinic lab has these, so you can measure. And if they're elevated, try shiitake or neem leaf, see if it reduces down to a normal level. But you can also measure the things produced by the regulatory T cells, because sometimes those are the things elevated and you might not catch an elevated level in the blood. And so you can measure TGF, transforming growth factor beta, which can be produced somewhat by the regulatory T cells and by other cells, and it's not a good thing to have elevated. And there's research done in Cancer 2001, the journal Cancer, bladder cancer patients, 51 that underwent surgery to remove their bladder because their bladder had a invasive type of bladder cancer, and they measured TGF before the operation, and they correlated it with the pathological features, how advanced was the cancer. And they found that patients that had the TGF elevated before the surgery had more of the invasive, invading the muscular wall of the bladder. And then they noticed that after the surgery, these patients were more likely to get a reoccurrence and had a shorter survival. Why? Because TGF prevents those killer cells from attacking. So again, you want to measure TGF. And I just mentioned earlier that Neem leaf has been shown to reduce that. Neem leaf, not neem seed oil or anything. The leaf extracts are something that can be used if a patient has an elevated TGF. And shiitake mycelium extracts, not the fruiting body, not the mushroom, but the mycelium. And then earlier I've talked about enzyme therapy for lowering TGF. Enzymes have not been studied, to my knowledge, on affecting regulatory T cells, but they have been studied to reduce TGF production, possibly by regulatory T cells. And there's enzymes found in pork pancreas that have been studied, and enzymes found in papaya called papain. And there's a mixture of these enzymes called wobenzyme that has been studied. Other things that can affect the regulatory T cells is there's this compound interleukin 10. Interleukin 10 is produced by a type of regulatory T cell that I mentioned, and so you want to measure interleukin 10 as well. Interleukin 10 can be lowered by herbs such as pau d'arco, and a form of vitamin E called tocotrienols. And so I talked about interleukin 10 in the last week's show, because it's also responsible for inducing the production of myeloid-derived suppressor cells, another suppressor cell that protects the cancer. So interleukin 10 can be measured in the blood. If it's elevated, those are some natural compounds that can be used to help reduce it so it doesn't lead to a suppressive effect. Another thing that can be measured that gives you an idea if there's a lot of regulatory T cells forming is something called IDO. Stands for this big name called indoleamine 2,3-dioxygenase, IDO for short. Well, IDO is produced by dendritic cells when they come in contact with the regulatory T cell. So the dendritic cells are in the tumor too. They're doing their inspection process like they're supposed to. And their goal is to come in contact with the T helper 1 cell and start some activation. Well, if those T regulatory cells are in there because the cancer brought them in, then the regulatory T cell grabs a hold of the dendritic cell and causes it to produce this thing called IDO. And what does IDO do? IDO breaks down something that's in the vicinity of these dendritic cells and these T cells called tryptophan. Tryptophan is another amino acid, just like I mentioned, arginine is another amino acid that's from a protein. So every time you eat protein, you get this thing called tryptophan, and it circulates all in our body, and so it will be in the vicinity of the cancer, surrounding it. And if IDO is produced in excess by the dendritic cell in response to the T regulatory cells, tryptophan gets broken down, and it gets broken down into something called kyurenic acid. And that actually leads to cell death of the T cell. And so T cells, the killer T cells that is, undergo cell death when there's all this kyurenic acid in their vicinity. So it's not necessarily that tryptophan is needed for the T cells, but that the formation of this tryptophan breakdown product induces cell death of our killer cells. And that's another way that the regulatory T cell protects the cancer cell from the killer cells. And and what's interesting about this is tryptophan, you might have heard of, is used to help make serotonin in our brains. Serotonin is a compound that makes us feel good, less depressed and everything. That's what, Prozac and Zoloft increase. So there's some research, that, people with cancer, can certainly get more depressed. But sometimes, even if, they're strong-willed and things are still good in their life, that they feel depressed for no reason in particular, and they can't snap out of it. And sometimes it's because the tryptophan in their body is being broken down by the cancer, and they can't make that serotonin that they need. So depression can be an effect of just the process of cancer, biochemical process, not necessarily just by going through the cancer. So you can measure this tryptophan breakdown product in the urine and in the plasma. And so you can measure this thing called kyurenic acid or kyurenine. And there's studies done in Spain in the Molecular Biology Reports 2013, people with colorectal cancer, noticing that those that had high levels of this kyurenine in the plasma, in the blood, had lower survival rates of 57 months versus those that had normal levels of kyurenic acid, 80 months survival versus 57 months. So another thing that can be measured, Genova Labs in North Carolina has that test, can be measured to determine if that's an elevated way. Another way of determining if the regulatory T cells are being produced. So you can measure that. You see elevated levels of kyurenic acid in the plasma. Then you can there's natural compounds that have been shown to reduce that. Resveratrol is a compound that you might have heard of. It's found in grapes. It's red wine and stuff has resveratrol. It's also found in chocolate and then other plants and stuff. And they, in a research done in mice, biochemical, biophysical research, Communications 2013, they gave mice a cancer of the thymus gland and gave them resveratrol or just a placebo, and they noticed a decreased level of this IDO, so they weren't able to form that kyurenine and weren't able to suppress the killer T cells. Well, they found that the tumors that had the IDO positive, that had those cells around it, their IDO positive, were 1/3 the size with the resveratrol after 40 days. But there was tumors that did not happen to have a lot of these regulatory T cells, and the resveratrol had no effect on them. So it's very specific if there's a high amount of this IDO being produced by the dendritic cell in response to the regulatory T cell, that resveratrol can be indicated. So if you measure that in the plasma, the kyurenine, see elevated levels, resveratrol is one choice. Neem has been studied for this. So neem is has many effects. It reduces the numbers of those regulatory T cells in the tumor, but also reduces the production of this IDO. And I mentioned it even increases the killer abilities of those T cells. So there's a study done in human immunology in 2013 in patients with cervical cancer. This was a human study. And they treated the regulatory cells and the dendritic cells from patients with cervical cancer with neem. So it was kind of done outside of the body, where they took these cells out of the blood of these patients with cervical cancer, gave them neem, and then looked to see what happened, or gave the cells the neem and incubated them together, and noticed that the production of IDO by the dendritic cells in response to the regulatory T cells was much lower. And then it also increased the production of IL-12 by those dendritic cells. So dendritic cells are really necessary. They produce the IL-12 and help with the activation of the T helper 1 cell and the killer cells. But if they are affected in any way by the regulatory T cells, then they produce this thing, the IDO, that breaks down the tryptophan that destroys the killer cells. So they can be manipulated by those regulatory T cells. Neem prevented them from being manipulated by those regulatory T cells, at least outside of the body when they incubated it together. But in an animal study, given neem together, it was shown to help reduce the numbers of regulatory T cells surrounding the cancer, reducing the suppressive compounds produced. So it's very interesting substance that really needs to be studied more, neem. Another type of suppressor cell is the T helper 2 cell. So we talked about myeloid-derived suppressor cells, neutrophils, cancer-associated fibroblasts, regulatory T cells, and then there is another type of suppressor cell called the T helper 2 cell. The T helper 2 cells are types of T cells that also produce immune suppressor compounds like IL-6, IL-10, and then something called IL-4 and IL-5. And we already talked a lot about a lot of those compounds and natural things. So these can be measured. You do a simple test where you take blood from a person with cancer, and you take out the T cells, and then you stimulate those T cells with different compounds, and you see what they produce. Do they produce a lot of things that we want? IL-2, gamma interferon, IL-12, or are they producing a lot of the things that have a suppressive effect? IL-6, IL-5, 4, IL-10s, IL-13s is different ones. And then so you can get an idea, is there a dominance here of T helper 2 cells, or is there more dominance T helper 1? We're always going to have T helper 2 cells. They help keep T helper 1s in check. We don't want a dominance of them. And there's all kinds of research showing that a lot of people with cancer do have a dominance of the T helper 2 cells. These assays, these blood tests show that there's a more predominance of those. American Journal of Surgery in 1999 took patients with colon cancer, and they measured they just did this, what's called a production assay before surgery, and they noticed that there was a higher amount of these T helper 2 cells, higher amounts of interleukin 6, interleukin 10, all that stuff being produced by the T cells. And then they took the tumors out, and then they remeasured and noticed that these T helper 2 cells declined. So the cancer cells are producing compounds that shift our T cells from tumor-fighting cells to tumor suppressor cells. And there are 2 compounds that have been studied for this. One is called Moducare, which is from pine trees. Moducare is a compound that called cytosterols and sterolins that have been found in pine, that have been studied, human studies, to shift T2 cells away from T2 to more T1 predominating. And then mistletoe, the same thing around Christmas time. Mistletoe has been lots of studies on that. And like in Germany, there's extracts of mistletoe, injectable forms of this plant mistletoe, and that is one thing. So you see elevated levels of T helper 2 cells, Moducare, and mistletoe can help. All right, we ran out of time. We're done with the suppressor cells. When I get back on next week, we're going to talk about killer cells and ways to make them kill cancer better. This is Dr. James Belanger. I'm your host with Cancer Concepts and Compliments.
Thank you for joining us this week. Cancer Concepts and Compliments with Dr. James Belanger can be heard live every Tuesday morning at 6 a.m. Pacific Time, 9 a.m. Eastern Time on the Voice America Health and Wellness Channel. We hope to see you next week for another show.